5 hours on Tuesday and 4 hours on Wednesday, right? So very very long talk, but so my lecture is just now I knew so intelligent biomedical technology and microfluidic 3R applications. Okay. So anyhow, so first I will introduce by myself. So I'm living in Tokyo, and my Tokyo Tech is also located in Tokyo or near Tokyo. And so distance between Tokyo and Taichung, not so far, but 3 hours on flight and 1 hour on the HSR. Then so in total around 5 or 6 hours, including some time. And then so this is our campus here. Maybe so this photo was very old, and so now I'm working here in the field here, and so this campus is out of Tokyo, so suburb of Tokyo or out of Tokyo. Then so we call nature-rich Suzukakedai campus of Tokyo Tech. And also so I forgot to give the acknowledge to them, and so first I introduce my laboratory, and so my laboratory is not so big, but then so she's from New Zealand, and he's from Norway, and he's from China, but he was born in Japan, and other persons are Japanese. And sometimes other country foreign students also came to my laboratory and to get the PhD. But I have never received Taiwanese students. So of course, so I'm waiting, okay, but I have never. So, by the way, so this lecture introduces microfluidic device as an alternative method of animal testing, and as shown here. And but even if I just introduced them, so you will not know what is so great or interesting about it. So firstly, so I have to explain background of this microfluidic device technology. In fact, the current state of development of a microfluidic device is that many of the things that can be done adequately with cell culture in a dish simply make smaller, and culture medium is poured over there. So it's I think most of microfluidic device technology have not so much advantage by themselves. Anyhow, so first, so in particular, it seems anyhow so difficult to understand how device development can replace animal testing, as many people are engineering, so especially process engineering, and so And also, so I majored in industrial chemistry before moving into biology. So actually, so I was graduated from the Department of Industrial Chemistry, University of Tokyo. But so I belong to the laboratory working on molecular biology. Then so after getting master degree, I moved to the Institute of Medical Science, University of Tokyo. Then so I started to study on developmental biology or developmental engineering to produce interferon gamma knockout mice and other transgenic mice. Then so at that time, so I really worked on the animal testing. And then so I have at that time so already I had experience using the embryonic stem cells. Then so I produced, of course, using this technology. So I produced the I produced the knockout mice and the transgenic mice. Anyhow, so I can operate embryonic stem cells. So in this class, I will, of course, I will explain what the embryonic stem cells are. But anyhow, so embryonic stem cells are pluripotent stem cells. Then to be differentiated to all of the cells of the body. Then so we call them pluripotent stem cells. Then so I changed to the alternative methods of animal testing by using the embryonic stem cells and human iPS cells and so on. Then so anyhow, so I was graduated from the industrial chemistry, and then so maybe I hope I can help bridge that gap. And so I will talk about the device on the last day, so Wednesday. But today and tomorrow, I will talk about the background, or rather than basic knowledge. Okay, rather the basic knowledge. And so maybe you have already received this schedule. So today I will talk about advantage and too small. Talk about advantages and the disadvantages of animal testing, and the need for alternative to animal testing, and organs and the cell culture, and also the importance of circadian rhythm. So we have a circadian rhythm. And also, how to immortalize the cells, especially the primary culture cells. Anyhow, so alternative animal testing methods are made possible by the result of many previous animal tests. So animal experiments are still very important. Then so we are now standing based on the animal testing result. And anyhow, so today are here. And firstly, animals would be classified in this way. So wild animals, pet animals, laboratory animals, and farm animals. So wild animals are born with the intention of living in the wild. So they are born to live freely. It's a wild animals. So they have a right. They have a right to live on the earth. And so pets are also born to be loved by people. And farm animals, on the other hand, are born and raised for people to eat. Then, so laboratory animals are born and bred to be used in experiments. So, just animal was classified by this thinking. And then so, but even so, laboratory animals have lives. Okay, so all animals have lives. So based on this idea here, so first, so let's think about about the laboratory mouse. So of course, laboratory mouse has also a life. So laboratory mouse is also subject to the animal welfare. So animal welfare now very, very important. So we have to think about it always. And then, as you know, the 3 R's are important in the experiments. So such as 3 R's, of course, replacement and reduction and refinement. So microfluidic device technology is so belonging to maybe the replacement of 3 R's. So of course sometimes and also sometimes associated with the refinement also, because we can use human cells also. Then so maybe I will explain later. And next, so second point. So human animal differences. So differences between the human and animal. So although both mice and humans are mammals, there are many differences. For example, I'm now a virologist, so I'll talk about virus infection. For example, HIV causes the AIDS in humans. So, of course, firstly, they try to infect the animals with HIV, but they fail because for example, a mouse has no receptor for HIV. Then, so HIV cannot infect mouse. Then, so mouse cannot become the HIV infection model, of course. And then, so we introduced the human CD4. Human CD4 is one of the receptor of HIV. Then, so we introduced the human CD4 expression vector into the mouse. Exactly, speaking, to produce the human CD4 transgenic mouse. But this transgenic mouse also were not HIV infection model. Then so this human CD4 molecule is not sufficient to give the HIV infection. Then so next we are looking for the second receptor, and then now we know the chemokine receptor is a second receptor for the HIV infection. But still, these transendomers cannot become the infection model for the drugs, anti-HIV drug screening, because the immune system is completely different from the human one. Then so anyhow, so difference between mice and humans are very, very big, and that's a big problem. And the second, because mice are smaller than other animals, so they take up less space and more economical to breed and maintain. So it's a very big advantage for laboratory mouse. And fourth, there are many inbred strains of experimental mice. So it's also a big advantage. It is easy to syngenically transfer, so implant to the same inbred mice. Okay. And then so this is also a major advantage for the laboratory mice. And fifth is also a major advantage for laboratory mouse, understand the complexity in individuals. So especially human immunity and mouse ones are big different, but of course some of them are very similar, and so laboratory mouse has a very big advantage to study on the immunity also. So in total, so except for animal welfare and a little bit of differences between the human and the animals, so laboratory mouse. Laboratory mouse has a very big advantage to study, and also elucidating to the human beings. And however, anyhow, so all animals have lives, and so we have to think about these issues. And by the way, you have heard that something is good or bad for your health. So so for example, coffee is very good for your health. But maybe some people are afraid. So coffee contains a lot of caffeine, and then so it works very bad. But anyhow, and also for example, lycopene, is also very good for your health. So tomato contains a lot of lycopene, and so it's good. So maybe you have heard some of them. But anyhow, so is that true? And I have heard that it's opposite. And also, does it affect me, or only young or old people? only for healthy people or something. And important points. Is there any evidence? So, what evidence would make you believe that? So, please think about it. So, so I will give the example. So, is it true that people who drink a lot of alcohol are more likely to develop lung cancer? So, can you believe that? So, strong alcohol drinking people have a very strong risk for the lung cancer. So actually, so there is a correlation between the so strong alcohol drinker and lung cancer. So there is a correlation already. So do you believe it? So first, I think was this trial have been done using the volunteer of the human people. So I don't know, I'm not sure there is a paper of animal testing, but actually, so there is a correlation between the strong alcohol drinker and incidence of lung cancer. So it's a very important. So I'm going to ask you on next slide. Okay. So it's a little bit answer. So I will ask all of you. Okay, first. So, what do you think? So first, my first question is, so do you believe this result? No. Why? Mm. But I think this experiment has been done very big population, maybe. So just 10 people, maybe for 100 people, or maybe, and then so they have many conditions. But anyhow, so they compare the alcohol drinking people group and so no drinking people, and they just compare. Then so strong alcohol drinking group have a very big frequency or incidence of lung cancer. So of course, non-drinking people. So some of the non-drinking people suffered from the lung cancer, but a small number. So, so if you are afraid of the lung cancer, so you maybe you don't drink alcohol, right? So not a good question. But okay, so maybe time is very important. Just so I asked everybody yes or no. So do you want? Do you believe this result? Yes or no, please. Yes. You? Yes. Oh. Yes. Yes or no? No. Maybe yes. Yes.
I think if we talking about this in a human or just in intelligent, I think it would be yes. But I'm not pretty sure about if we put the alcohol to contribute the cell line. I'm not pretty sure it would to become the lung cancer. Because as I know that the liver have we were to distinguish two groups about the non-alcohol disease and another one is the alcohol. So it is very interesting that the alcohol into the liver will release some of the metabolites to induce the lung cancer. And another opinion from my experience, and I had to do a meta-analysis to estimate about the lung cancer's patients. The 16S microbiota from the gut and oral, and also the lung cancers, they will have associated, you will have a correlations that the 16S microbiota will to facilitate the lung cancer progress from the first stage to the fourth stage. So it's going to be very interesting that the liver microbiota and also the gut microbiota and the lung cancer.
Very good, good discussion. But in this case, anyhow, so that's a result, correlation.
I think it is true. Because, but there's no strong and very direct evidence.
Okay. You?
Yeah, I think so, but it's not totally persuasive because it's only a phenomenon, and maybe there's something else to cause the lung cancer, like the people who like to drink alcohol, they have some similar habits in their daily life. So, and maybe those their habits cause the lung cancer or make the lung cancer to be more severe. So, I think there have to be evidence to persuade
So it's a really way to talk about it. Okay. And then?
Because I don't have more information for this, so I don't think it's true.
Okay, yeah, so you need more information, yes. So, correlation does not imply causation. So causation is necessary but not sufficient condition. So this is a correlation example, but causation is a smaller area than so like this. So correlation does not imply causation. So there is a so we call co-founding factors, as you told. So first one is affects outcome. The second one associated with the factor. And the third one, not an intermediate factor between the factors and the outcomes. So in this case, so there is a correlation between heavy drinkers, heavy alcohol drinkers, so frequent drinkers groups, and high incidence of lung cancer. So there is a correlation, but it's so really, yeah, of course, but it's a wrong consideration because like this. So most of heavy drinkers or frequent drinkers prefer smoking. That's also a correlation between heavy drinkers and the high rate of smoking. And then, so also a strong correlation between the high rate of smoking and high incidence of lung cancer. So animal experiments suggested or indicate already that smoking causes lung cancer. It's a scientific evidence already. Then, so maybe, so of course, liver contributes or something or something. So it's so more deep consideration. But anyhow, there is a very strong and scientific correlation or causation. Smoking induces development of lung cancer. So now this way is very important and necessary. And then so we call this one confounding factors. So it's a smoking. Okay. Then so we have to think about confounding factors. And of course, participants are randomly classified as smokers or non-smokers and not subjected to experimental intervention. So there are many cohort studies, cell biological experiments and animal experiments and so on. Then anyhow, so in this trial, it was not so good because they didn't consider confounding factors such as smoking population and so on. So and then so now randomized control trial is very very important. And so we have to think about correlation between A and B. So we have before getting the result. First, we have to think about a randomized control trial. So including the confounding factors. So what in this case, what are the confounding factor, and so on. And then so Yeah, it's okay. And then so also, so there is there are different levels of evidence. So you see, so bottom, animal and laboratory study. So cell culture study also included here. And then so case report or case series, narrative reviews and expert opinions and editorials. So these comments or results, of course, should be standing on the animal or cell culture study. Okay, and case control studies. There are many reports, and a cohort study here. And then so randomized control trial here. And then so based on the randomized control trial, then so meta-analysis, systematic review should be carried out here. And then finally, going to can go to the clinical practice. And also we have to make the guidelines, so based on the bottom of these trials or experiments. So, the higher the level of evidence, the more reliable it is than this. Okay, so cell culture. So alternative of animal experiment and also animal experiments. So at the bottom. So not so in this case so not so reliable, but in this case so just in terms of the clinical application in humans. So animal experiments, I mean, it's okay, but so we have to elucidate the human being phenomenon. And then so we need more studies, a very long way to reach the clinical practice. And so it's a very important. But of course animal experiment or alternative method of animal experiments, cell culture or microfluidic device technology also now just bottom of this pyramid. So anyhow, so RCT, randomized control trial, is very hard. So before starting the randomized control trials, the bottom line is the importance of the results of animal and culture cell experiment. Okay, so it's just also background to consider the animal experiment alternative. So considering the alternative of animal testing. It's a very animal experiment and also the alternative of animal testing. They're standing just the bottom of this pyramid, but very very important because before starting before our randomized control trial or other cohort study also starting, they need enough data from the animal experiments or alternative of animal testing. So next talk I'll talk about animal organ versus general cell culture. So this is the main major points of my talk. on my lecture. So first one, cells in organs. Cells in organs are three-dimensional. I will explain later. But cultured cells are generally flat. So it's a morphology. Okay. So have you ever cultured mammalian cells? Raise your hand. Oh, only two. Okay. So and also I gave you the technical term list. So if and then sometimes confusing the technical terms in this field. For example, so in vivo and but I didn't describe the in vivo and the in vitro. So in vitro, in our field, in vitro indicates the cell culture experiment in our field. But in the bacteriology field, maybe bacteria is a cell, and then in vivo experiment. So in vitro just in testing tube or something. But in our field, so in vivo means animal experiments. And then cell culture experiments, just in vitro experiment. And culture cells, of course, in vitro experiment, generally flat. Next, second one, cells in organs. Cells in organ means real cell in organ. So hepatocyte in the liver. Okay, not cultured hepatocyte. And cells in organs adhere to something on most surface. Whereas cultured cells adhere only to the bottom surface. It's a simple cultural system. And the organs are composed of multiple cell types. So I will explain later also. Anyhow, liver consists of not only the liver cells, so hepatocytes, and also sinusoidal endothelial cells, and and also cholangiocytes and so on. And anyhow, but the cultured cells are generally a single type because depending on the extracellular matrices, then one cell preferred the collagen-coated dish, but on the other hand, another cells prefer the hyaluronan or other matrix. Then it's quite difficult to co-culture all of them. And next one, cells in organ may have different physiological functions upstream and downstream of blood vessels. So maybe I will try to explain later regarding the hepatic tissue, so liver tissue. But cell cultures are uniform because cell culture system has no blood circulating. And organ cells have uniform circadian rhythm, but the cultured cells have a different circadian rhythm. So that overall there appears to be no circadian rhythm. Maybe this later I will explain also. And also it's difficult to culture cells as normal cells of organ origin. So it's quite difficult to maintain the physiological functions, real physiological function in the cultured cell. And blood, of course, blood circulation cannot be reproduced in culture systems. And that's why we invented the microfluidic device. Because the microfluidic device, of course, can make the blood circulation system also. And also the coexistence of indigenous bacteria cannot be achieved in the culture system at the moment. But of course, now we succeeded to co-culture system of intestinal epithelial cells and intestinal bacteria. Okay. Anyhow, so those are limitations of our general cultured systems. So I added the general because we invented the novel culture system. So novel culture cell system by using the microfluidic device. Okay. And then now maybe in our I will explain these blue sentences here. Okay. Maybe I'm sorry. Maybe you have already my explanation before, so previous lecture, and then so but almost the same. But maybe so you are clever, but you will forget something. So this is a liver. You know, human liver. And this is the front view of the liver. So liver is very big here, completely occupied in this space. So very big. And the liver plays very, very important roles in metabolism and toxification and accumulation of the blood and also so detoxification and also producing many types of the proteins and so on. And then so here you see, so right lobe is much bigger than the left lobe here. So shape is it's different. So shape in human liver completely different from the mouse liver shape. Mouse liver has six lobes, and so shape is also different. And then, but in human, so liver, human liver has two lobes. Right lobe and left lobe here. And so here is the artery. Artery is very important to deliver the solid oxygen. So anyhow, so deliver oxygen by the erythrocytes. And here, portal vein. Portal vein, it's completely bigger than the artery. So most of the blood supply by this portal vein. So portal vein connected to the intestine. So of course much of nutrients and molecules they are incorporated at the intestine, and then these molecules deliver to the liver directly via portal vein. Okay, so portal vein is very important. And this is a common biotype and just also so reverse direction of a flow to the liver to intestine. Okay, and also you can see the gallbladder here, but this is the liver from the back. So you find the gallbladder here. So gallbladder here. And this is a vena cava, very big blood vessels. Also, and the artery and the portal vein here, like this. So mouse also are almost the same style. So mouse has also a gallbladder, but on the other hand, rat is a little bit just bigger than the mouse, but the rat has no gallbladder. So different, and the horse also. Horse has no gallbladder, and maybe so. You are now belonging to a veterinarian course, and maybe you know well, maybe better than me. And here, so just a cross section of a human liver. So this is a hepatic lobule here, and hepatic lobule, and here central vein and the portal vein here. And so let's look at one hepatic lobule here, this one. So this is the hepatic lobule, and here the portal vein, and the red tube, smaller red tube is the hepatic artery. So blood flow direction from here to the inside of the hepatic lobule, and the bile duct here. Bile duct flow direction is opposite. Okay, and then so blood supplies from the portal vein or hepatic artery, and then to so central vein here. So this is a cross-section schema of the liver lobule. The portal vein here. So portal vein comes from the intestine. I told you, and small molecules containing the nutrients and the toxins and the other molecules and so on, and flow into the liver lobule here through the sinusoid here. So sinusoid. So and also hepatic artery joins the portal vein and becomes a anyhow. So portal vein and the hepatic artery join, and then to make the sinusoid, okay, flow into the liver lobule here and to central vein. So that's a very important point for this lecture, because we made this hepatic lobule structure on the culture system, using the microfluidic device. Then so first you have to understand the structure of the liver tissue like this, and then so look here. So here, so this is the endocellular, so sinusoidal endocellular cells. So sinusoidal endocellular cells are fenestrated. Fenestrated means so having the many windows, so many holes like this. So compared to the other vessels, so wall of the vessel is very strong. But on the other hand, the so sinusoidal vessels, so sinusoidal has a very weak wall of that. And then so because so you can find many holes like this, very weak. And that's why nutrients and other so molecules can infiltrate into the liver or hepatocyte, like this. So this sinusoid. Sinusoid means like a vessel. So sinusoid is not a blood vessel, like a vessel, because a very weak wall. Because they have a very weak wall, and so sinusoid constructed by the endothelial cells. So we call sinusoidal endothelial cells. Okay, and also here hepatocyte exists here. Okay, and so I will explain in detail here. So yellow ones are hepatocytes. Okay, then so green ones and so sinusoidal endothelial cells. So hepatocytes firstly face to the sinusoid here, but hepatocytes do not adhere directly to the endothelial cells. So there is a distance space exist here, so containing the much extracellular matrices. Okay, and then anyhow, so here, so this face, the face to the sinusoidal here, we call this face a basal or sinusoidal face. Okay, so generally we call a basal face here. Okay, and here gray area, it's biocanalicula. So biocanalicula connect to the bile duct. So biocanalicula is so bile duct, but a very small one. So this face, okay, this face we call apical face or biocanalicula face. So generally, so apical face. Okay, and also hepatocyte adhere to the neighbor hepatocyte. So just so here we call lateral face. Okay. So hepatocyte has three faces at least: basal, apical, and lateral. Okay. So and also I forgot to tell you. So hepatocyte shape of the hepatocyte is not flat. Okay, just a very 3D structure on the 3D structure, and then so also having the three faces, including the basal, apical, and lateral. It's very important. So apical is face to the outside. Okay, outside. Because this biocanalicula goes to the bile duct, and then bile duct goes to the intestine. So inside of the intestine is the outside. So this face is to the outside. That's why we call apical. And the basal means basal face to the inside of the body. Inside means real inside. Anyway, so basal face. Okay, and anyway, so okay, and this one, oh, it's the same maybe. And so basal face, so here, hepatocyte adhere to the matrices. Okay, this space is not a cavity, so there are some matrices. Okay. And so anyhow, so hepatocytes adhere to the matrices, some matrices. So cell matrix adheres, and also this face. So just so face to the biocanalicula. And so anyhow, so and also the third one is hepatocyte face to the hepatocyte each other. Exactly speaking, hepatocyte adhesion. So cell adhesion mainly a cell matrix and cell adhesion. Both. And so this slide shows general structure of the epithelial tissue structure. So here is so epithelium, and you can find of course epithelial cells, and also you can find fibroblasts and so on, and also here basal lamina consists of extracellular matrices, and also collagen fiber and elastic fiber and hyaluron or proteoglycan and so on. It contains a variety of cells and extracellular matrices. And it contains a variety of cells and extracellular matrices, and also maybe, I don't know, okay. And by the way, so here, the surface of the up on this exterior, it's free, okay? And but other sites adhering to the neighboring cells, and also adhering to the basal lamina here, and so on. And anyhow, so cells need to attach to something to survive. So even the lymphocytes. So lymphocytes, maybe you believe they're free, but the lymphocytes also rolling, like adhering the vessel wall, thing like that. And then so this so this is a primary hepatocyte culture on the collagen coated dish. You see, so primary hepatocytes were prepared from the mouse liver, in this case, by the perfusion method, collagenase perfusion method. And in general, it has been still difficult to culture primary hepatocyte for a long time. But anyway, now these hepatocytes are cultured on collagen-treated dish, which partially mimics liver tissue. But unfortunately, it's a completely different microenvironment between liver and the culture system. Anyway, so at the bottom, the cells adhere to the collagen, and sometimes the side of the cells adhere to the neighboring cells, but the top of the cell is free, so face to the medium. And also shape is flat. It's completely different from the structure of the hepatic tissue, like this. So that's a very big problem. So maybe we don't believe the cell culture system cannot become the alternative of the animal testing, because so many different style, structure, and also cell cultured cells have very low physiological activities and so on. And but anyhow, so most of you have never experienced to culture the cell, mammalian cells, and then so I will try to explain a little bit about mammalian cell culture. So here, so typical mammalian cell, looking at horizontal section. So there are many types of organelles, such as mitochondrion, Golgi apparatus, plasma membrane, of course, and the nucleus, and endoplasmic reticulum, and the lysosome, and so on. So maybe you have learned already. It's just a cultured cell. And so the cell in the organs shape is completely different, of course. And also mammalian cells cultured under this condition. This condition means so 5% CO2, and also humidity is also very necessary. And mammalian cells are cultured on the dish generally like this. Okay. And of course, mammalian cells needs medium. Completely depends. So composition of a medium is completely different in the several mammalian cell types, and also the scaffold and how to dissociate, and the timing of passage are completely different, depending on the types of the mammalian cells, like this. And also there are many types of media. So just I introduced some of the cell culture media like this. DMEM and local DMEM and RPMI 1640 and IMDM and alpha-MEM and Williams and so on. And so we have to choose one medium for the one cell types. For example, DMEM. So direct cause modified ego medium we call DMEM. and MEM is a minimum essential media, and IMDM is modified direct coding media. Then so when we culture primary hepatocyte or hepatic tissue, we use Williams E or IMDM containing the FBS or something. But other cells, for example, fibroblast, maybe to culture the fibroblast, we use DMEM containing the FBS or something. So there are many types of the medium. But generally, for example, DMEM contains 15 amino acids and vitamins and inorganic salts and glucose and also other things. So you see, so glucose is very important because in our body, oxygen is always delivered to the peripheral sites by carrying, by erythrocytes. So it's a very clever, smart technique. So once erythrocyte receive the oxygen at the lung, and then so this erythrocyte deliver the oxygen to the peripheral, and then so maybe and then so return to the lung again. Then so it's a very smart system. But on the other hand, the culture system. So in the culture system, there is no erythrocyte. Then so, but of course, culture cells have to survive, but no oxygen. Then so culture cells have to generate energy to survive. Then so you have already learned aerobic metabolism and anaerobic metabolism. Well, firstly, so glucose dialysis, then so glucolysis to generate the ATP without oxygen, under the anaerobic condition. But to make the much of energy, so we need aerobic metabolism, beta oxidation, and also phosphorylation, so electron phosphorylation system, and so and yeah. So, but in this case means so cell culturing system. So it's quite difficult to supply the oxygen. Then so generally in the cell culturing system, it's anaerobic. Then so we have to supply the much glucose. Then so to make the ATP, okay, by the glycolysis. So it's I think next maybe I have just a confirmation. So do you understand how to culture mammalian cells? Is it okay? So most of you have never experienced to culture mammalian cells. And but unfortunately, so it's very difficult to culture mammalian cells because generally we can culture mammalian cells, but most of the mammalian cells are cell lines. Cell line means cell lines. Most of cell lines are established from the tumor. Then that's why we can culture the mammalian cells. But so tumor cells, it's a cancer cell. It's completely different from the normal cells. And then so now we are thinking to establish the alternative method of animal testing. So we need firstly normal cells. And so it's quite difficult. And so now, so maybe so it's still difficult to culture the primary cells. To of course, just culture it's okay, but it's quite difficult to keep the physiological functions in the culture system. And then so now and then so next term so I will explain another cell type. Just a moment. So cells in just yeah. So yeah, maybe tomorrow I will talk about human iPS cells and also embryonic stem cells, and it's a little bit similar to the general tissue of the organs. And then so, but anyhow, it's quite difficult. And so next maybe let's have a break 10 minutes, and then so restart 10:30. Okay. Then no more than this one. Okay. Let's have a break. So next, I'll talk about circadian rhythm in the individual, and also we are now trying to introduce circadian rhythm in the cell culture system also. It's very important for alternative establishment of alternative of animal testing or human test. And then so anyhow, so laboratory animals are kept in well-controlled day and night lighting cycle. So so by the way, so I don't know. So only two of you have experience of mammalian cell culture. And so I have a question. So do you have experience of animal experiment? Yes, animal experiment.
Not really.
Not really. Oh, so most of you have no experience of animal experiments, right? So maybe so you don't so I mean you don't have experience for taking care of laboratory animals. So you don't have. So maybe so I will try to explain anyway. So anyhow, so maybe neighbor building. So you have animal facility, right? Maybe. Yeah. And then so anyhow, so laboratory animals, so including a mouse and a rat and a rabbit and so on. So they are kept in a well control day and night lighting cycle. Maybe I'm not sure. In my institute, maybe at 8 a.m. Light is turned on, and then 8 p.m. Turned on. It's a periodical. It's very important. And then so now I have one hour time difference between Japan and Taiwan, and now it's okay. But in the night, maybe around 11 p.m. and then so I little bit sleep. I'm a little bit sleepy. And then, but anyhow, so laboratory animals are maintained under the very fine control. Not only the light cycling, lighting cycle, and also temperature and humidity also. It's very important. And and so they therefore have a circadian rhythm, just like us. Maybe more. They have so they have more. They have maintained under the more strict lighting cycle. Anyway, so and so well, but the impact mice do not have the same precise circadian rhythm as humans. So maybe so humans, most of us, so in the midnight we sleep, and daytime we waking now, like now. But in the mouse, so mice has a not so good circadian rhythm exactly. So because they are really afraid of their enemies. So they are so wild mice, of course, wild mice have a very dangerous enemies in the field, and then so they maybe they can't sleep well, and then that's why so they have not so good, not so strict circadian rhythm exactly. But anyway, so that's also the difference between the human and the laboratory animals. But anyhow, so now they have also the same circadian rhythm as us, but also a little bit. So I'm a little bit so afraid of you, confusing. So writing, but generally, mouse is very active in the night, and the daytime they are trying to sleep. So circadian rhythm is completely opposite to the human being. Human being circadian rhythm. Okay, it's a little bit. So it's a big difference. So they are always active in the midnight. maybe you have never sounds from the mouse or rat in your house, maybe, so no. But when I was a PhD student, so I live alone, so a little bit apart, so depart from the parents' house. And then so at that time, so I was not so rich, and then so I lived a very cheap apartment. And then so sometimes I have heard the sounds of the maybe not mouse, so rat. From the ceiling, and then so they are always so active in the midnight. Okay, and anyhow, so it's a little bit different. And also, of course, on the other hand, cultural cell has each cell has internal clock exactly, but the total, so in total, so system as a system, so cell culture system has no circadian rhythm, because they have, so anyhow, so cell cannot cell does not have cells have not do not have eyes. So we have eyes, and then so we can recognize now daytime or midnight or something. And so you know, so cells have an internal clock. However, for the body clock to function, so it needs command from the center here. So maybe so we recognize the sunlight here, and then so this signal delivered sent to the central nervous system. Exactly, so mainly so I don't know the exact pronunciation. So suprachiasmatic nuclear, suprachiasmatic nuclear. So anyhow, so we call it SCN. So I don't know the exact pronunciation. Suprachiasmatic nuclear. So SCN. So I call it SCN anyhow. So and so anyway, so. We perceive sunlight with our eyes, and signals are transmitted to the center. That is SCN, like this. And then so SCN sends a reset signal to the internal clock throughout. So whole the body, synchronizing the body's entire clock. Okay, so reset. So time is also same like this. That's why we have a 24-hour cycle called circadian rhythm, which regulate physiological activities such as sleeping and waking and so on, like this. So anyhow, so each cell have an internal clock, but all of the cells need a central signal to reset and to adjust the time. Okay. So just a moment. And then so let's look at the details of a circadian rhythm mechanism. So our main signal molecule is glutamate here, glutamate. So glutamate is recognized by the ionotropic glutamate receptors. Okay, receptors. Now, once glutamate is recognized by the ionotropic glutamate receptor, so calcium ion flow in, and in the calcium ion dependent phosphatase are activated, like here, activated. Okay, and then this active phosphatase. Phosphorylase, phosphorylase, so kinase, okay? So kinase, this activated kinase phosphorylate to the CREB. CREB is a cyclic AMP response element binding protein. So CREB, we call CREB. So CREB is phosphorylated, and then so phosphorylated CREB can enter into the nucleus here, and then so activated CREB can bind. So bind the CREB. CREB is a cyclic AMP response element of genomic DNA here. And just a moment. Why leave it there? Just a moment. Just a moment. This is not working. The computer went back to it's here. Thank you. Doesn't work. No card. It's okay. Okay. Yeah. I forgot. So anyhow, so activated, so phosphorylated CREB enter into the nucleus, and then so phosphorylated CREB bind CREB, so cyclic AMP response element sequence of the genomic DNA. So this element, it's like an enhancer or promoter of PAR1. PAR1 is a periodical gene, PER, periodical gene, so PER1. So PAR1 is a key molecule to control the PAR1 protein. PAR1 protein is a key molecule to control the circadian rhythm. Okay. Then so after the activation of CREB, and then so PAR1 expression of PAR1 is enhanced or so activated. Okay. Then so there are many molecules related with circadian rhythm regulation. So first PAR1, and another one is of course PAR2, PAR3 is almost the same group. and cry1, cry2 also. And then so PAR1 protein and cry protein bind each other to form the heterodimer here. And the clock protein and the BMAR1 protein to form the heterodimer also. Okay. Then maybe clock protein and the BMAR1 protein bind each other to form the heterodimer like this. Clock BMAR1, BMAR. And this heterodimer recognize the E-box and the E-box and bind to the E-box. And then so enhance the expression of PAR and cry expression, cry gene expression. And then PAR and the cry protein form the heterodimer. This heterodimer, PAR and cry heterodimer, replace the activation of clock BMAR1 heterodimer. And then this enhancement decreased. Then so PAR and cry molecules disappear. Then clock and BMAR activate again like this. So PAR1 and BMAR replace each other. So PAR replace the BMAR function. Then, but the BMAR enhance the PAR expression. And then so to make the rhizome like this. Okay? And then like this. So PAR1, BMAR increase, and then but PAR1 decrease. Then so like this. Okay. Then so three people who have been very active in understanding the mechanisms of these circadian rhythms molecularly. Then so were awarded the Nobel Prize in 2017. Like this. Not so recent. And so this circadian rhythm is not only the circadian rhythm related molecules, also this circadian rhythm affect the other physiological activities. For example, so metabolic enzymes also depend on the circadian rhythm. So this means that the pharmacokinetics of a drug are very different when there are more or fewer of these metabolic enzymes in the body, and the effects of the drug are different. Okay, so here, so metabolic enzymes, circadian rhythm, circadian rhythm here. So if the one drug, so if the drug administered at this point, so also at this point, metabolic enzyme also concentration of a metabolic enzyme also are very high, and then so this drug receive the metabolism by this enzyme. So it means become inactive. Okay, inactive. Inactive means maybe I will explain later anyhow. So inactive. On the other hand, here, sorry, so now, so I talk this one. So here, so so metabolic enzyme concentration is very high, so maximum. At this point, drug would be administered here. Then, so drug immediately would be immediately excreted. Excreted means so inactivated dramatically. So it means also no function. Then, but on the other hand, here, so metabolic enzyme level at the minimum here. At this point, drug would be administered at this point. And then, so this most of the drug can affect the effect on the some targets. Okay, so because most of the drug can maintain the activity, here. Okay. So I draw the mouse, but so human case also same. And so here we have animal experiments at the moment at this time. So we checked circadian rhythm related gene CRY. So CRY bind with periodical protein, and then so repress the activation of clock and bimal heterodimer. And then so anyhow, so CRY gene expression is lower, much lower at 10 p.m. Than at 10 a.m. And then so much. So CRY expression is much lower at 10 a.m. On the other hand, but rather than compared to the CRY expression at 10 p.m. Okay, so 10 p.m. at 10 a.m. CRY expression is very low. But at 10 p.m. CRY expression very highest here, like this. Okay. And but on the other hand, CYP2E1. CYP2E1 is enzyme, metabolic enzyme gene. It's one of the representative metabolic enzyme here. So this is acetaminophen metabolic schema. So this is acetaminophen. Acetaminophen is a core drug. So to remove the pain, to remove the pain, and to decrease the body temperature. So if you have a cold, you have a fever, and sometimes headache and then so after then taking this acetaminophen, then so fever dramatically decreasing, and also can remove the headache also. So acetaminophen is very popular in the world. Maybe, I don't know the Taiwan drug, but maybe this cold drug maybe contain the acetaminophen or ibuprofen or something. And so acetaminophen here, then normally after taking the acetaminophen, then so incorporated at the intestine and then so delivered to the liver. Then so most of the acetaminophen receive the sulfate or glucuronide conjugation, and then to excrete the outside. Okay, and but if you take the excess acetaminophen, and then so sulfate and glucuronide saturated, and then so other molecules. So glutathione also conjugated with acetaminophen and then to excrete outside, but also excess, and then so CYP2E1. So CYP2E1. So after the so if the sulfate and the glucuronide will be saturated, and then so CYP2E1 oxidize the acetoaminofen to the NAPQI, so N-acetyl-P benzoquinone imine. This so NAPQI is a very radical. Radical is means bind covalently to all proteins in a cell. So here now or here in the liver, so hepatocyte, and then so if the NAPQI occurred from the acetaminophen via CYP2E1, then so NAPQI bind to the all protein in the hepatocyte. Now, of course, it's a toxic, and the hepatocyte will die, and then so causes liver injury here. So CYP2E1 sometimes plays a good role, but sometimes plays a very bad role, like this. Then anyhow, so CYP2E1 expression pattern completely depends on the circadian rhythm. And then so when the cry expression is low at 10 a.m., the CYP2E1 expression is also lower. On the other hand, at 10 p.m., CYP2E1 expression is the highest, like this. So we inoculate, we administrated the acetaminophen into the mouse at each time, like this. Then, so you see, so at the 10 a.m., CYP2E1 expression is the lowest. And then, so you can imagine the low concentration of NAPQI because no CYP2E1 enzyme. And then, so acetaminophen is not oxidized by the CYP2E1. On the other hand, at 10 p.m., acetaminophen was administered, and then so NAPQI occurred from the oxidation of acetaminophen. Then so this NAPQI coated the liver injury here, like this. So I have to explain this one. So AST and ALT are deviating enzyme. Do you know deviating enzyme? Maybe no. So AST and ALT are abundant in the liver cells. Then so when the liver cells die, and then so the cells are burst. Then so broken. And then so inside AST and ALT is released. AST are released to the blood. And then so we call these enzymes deviating enzymes. It's those are marker of for the disease. So for example, AST is mainly exist in the hepatocyte, and ALT mostly exist in the liver cells, and also cardiomyocyte, so heart. And then so AST level is very so increasing. So medical doctor maybe diagnose it's maybe so liver disease. But ALT level higher, and then so maybe medical doctor diagnose liver disease or cardiac problem or something. But AST is higher, or this maybe so these data totally suggest the liver disease. But if the AST level is lower, but on the other hand, ALT level is higher, maybe not in the liver. So maybe this patient has a cardiac problem, or other tissues also. But anyhow, so these data suggest the liver injury in the mouse received acetaminophen like this. So maybe so when you take some drug, maybe you have never considered which time is the best for us. But this is a suggestion. But unfortunately, this is an animal case, so mouse case. So human means a little bit, or maybe the opposite. But of course, after, so before selling these drugs, so pharmaceutical companies always checking these pharmacokinetics. And then anyhow, so laboratory animals have a very big advantage to have a circadian rhythm. It's quite difficult to mimic the circadian rhythm in the cultured cells. Okay? So, so we have a circadian rhythm because we have an eye, and we have a circadian rhythm environment. Then so we recognize now it's daytime or nighttime. Okay, and then so this signal, so sunlight signal, sent to the SCN, and the SCN send the signal to reset into the whole tissue, whole cells in the body. But unfortunately, each cell, of course, cannot recognize the sunlight, and then so. But each cells have internal clock exactly, but so then they don't know how to receive the signal from the sunlight. Then so in total, in the total system, there is no circadian rhythm like this. So it's a randomized internal clock like this. So it means here, after the reset signal, receiving the reset signaling here, and then so circadian rhythm, all circadian rhythm in the all cells in the body, synchronized like this. On the other hand, in the cultured system, cell cultured system, so each cells have a one circadian rhythm like this, but in total, completely flat, to become flat. So it means no circadian rhythm. And then so anyhow, so existing safety evaluation studies in cultured cells. So time of preparation of a primary cell culture is not stated. So so it's so existing safety evaluation studies means how can I say so we can get the primary liver cells from the company. So primary liver cells were prepared from the human liver. But you can imagine, so this liver, of course, came from the human being. So this human being, of course, so human being needs liver. So, but the company can get the liver. So it means that this recipient has already died. So and then but we don't know what time the patient died. Then so we don't know the circadian rhythm of in this human being. Then so time of preparation of a primary cell culture is not stated, and the explanation is seed, and then so we don't know. That's a big problem. And also cell culture systems are not as receptive to light as an individual, and not all cells can be synchronized. Okay? And then so existing safety studies have not taken a circadian rhythm into account. So in other words, so they are not alternative method for animal or human. Then it's quite difficult anyhow. And then so in general culture cells, no toxic or medicinal effects are found. But in the culture system with a circadian rhythm, toxic or medicinal effects can be found. So I mean, for example, here, so this level is a threshold level. The threshold level means over the threshold level, we can recognize effective or toxic. So, but under the threshold level, we can't recognize the toxic or effective. Okay, and then so in general cell culture system. So non-circadian rhythm, and then so unfortunately at this time, so the average of the level is under the threshold level here, like this. So conclusion, in this conclusion, in the case of this experiment, the conclusion would be non-toxic or medicinal effect. Conclusion. On the other hand, if we have a circadian rhythm, if we have a cell culture system with circadian rhythm, so circadian rhythm is like this. So activity level is periodical changing like this. Then, so if you add the drug at this point, at the maximum point, and then so we can detect toxic level or toxic or medicinal effect like this here. Of course, at this point we failed to detect it, but maybe so we can add the drug now and also after the 12 hours, and we can detect one of them like this. Okay, and then so I think we have to establish the cell culture system with the circadian rhythm. It's very important for the purpose of the alternative method of animal testing or human testing. Then, so already we have information of circadian rhythm resetting mechanism. Then, so I have already explained. So cyclic AMP has a very important role to reset the signal of the circadian rhythm. Then, so phosphorine is a chemical molecule. This one activates the AC, and then so AC is cyclic AMP produce enzyme, and then so activate, and then so to produce the cyclic AMP from the ATP. Okay, and then cyclic AMP bind to the pKa, protein kinase A, and then protein kinase A phosphorylate and the ARC protein, and then deliver to the RSK, and then finally CREB, and then so CREB phosphorylated CREB bind the CREB sequence, and then to induce the power expression. Then, so we try this one first. So tomorrow I'm going to explain how to differentiate embryonic stem cells to cardiomyocytes. But now, so just please please know already. So we have a system to the differentiation of ES cells to the cardiomyocytes here, and then so we have ES cell derived cardiac tissue, and then so normally in the culture system no circadian rhythm, but we added the phosphorine here first. And then here again 24 hours later again, so 46, and then 70, and then after the 24, and so we succeeded to make the circadian rhythm in the ESL-derived cardiac tissues like this. You see? Honestly speaking, so we added the pulse scoring before 46 hours, of course, at the zero, and we added the pulse scoring at 24 hours each, and continuously, and then finally, so we succeeded to establish this periodical expression profile after the 46 hours. Okay. And also this is just so just a full screen exposure is just for 30 minutes. Okay, 30 minutes, only 30 minutes. Then so 30 minutes exposure of a full screen, and then remove the full screen, and then so we added after 24 hours again, like this. So of course, can you imagine this work? It's was very hard because so Sai Chen, he's a Chinese. At that time, he was a Chinese PhD student in my laboratory, and then so he added in total 154 hours. I don't know, maybe 1, 2, 3, 4, 5, 6, 7. So one week. So he had stayed in my laboratory in in a week totally. So and then he took the samples here and here. So he couldn't go home in a week at that time. So after getting this data, I knew. So why you can survive? And so so and then he answered. He brought his special bed into our laboratory. I didn't know that, but anyhow, so he got the data and he succeeded. And then also, so he checked. So this one is just power expression pattern, and also he checked other circadian rhythm related genes such as CRY1, BMAR1, and USB2. And you see the CRY1 and PAR1 is almost the same profile, but BMAR1 is a little bit covered. And but unfortunately, force scoring induces the cyclic AMP production. So cyclic AMP has a wide cyclic AMP plays a very wide range, not only to make the circadian rhythm, also to induce some other physiological activity also. And then so it's just we check the adjustment of circadian rhythm, but we couldn't check the other physiological activity because so pulse scoring has a side effects. Then so we invented other system. So this one is collaborated with Professor Imanishi at the Kyoto University. Then so she invented this fusion proteins, And this one is a little bit difficult, but anyhow, so DD is destabilizing domain, and ZF is a zinc finger. It's a very important, so necessary sequence for the specific binding activity. And NLS is a nuclear localization sequence, and AD is a transcription activation domain. It's a main domain of this molecule target. So it's we call artificial transcription factor. Okay, and so DD recognize the sealed one molecule. So after addition of a sealed one bind to the DD domain, so DD, and then so this molecule will be activated to bind upstream of a PAR1 gene to induce the expression of a PAR1 like this. Okay, then so degradation. Okay, so see, just a moment. I'm a little bit misunderstanding. So just a moment, because So this gene, of course, this expression vector is introduced into the mouse embryonic stem cells or human iPS cells. Okay, and then this expression is a expression vector. This gene is expressed constitutively. So constitutively is a expression is any type, okay? And then, but without sealed one, this molecule is immediately degraded. So disrupted, okay? Disrupted. But in the presence of the sealed one, so sealed one bind this site, then to stabilize this molecule like this. Then so this molecule can bind here, then to induce the expression of a PAR1. Okay. Then so we introduced this ATF expression vector into the mouse mouse embryonic stem cells like this by the electroporation. Maybe I will explain tomorrow how to introduce the introduce the gene expression vector into the cells. Anyhow, so by electroporation. And then so screening, and then we succeeded to pick up the colony expressing constitutively expressing this ATF gene. Okay, like this. And then so firstly, so no, of course we had to confirm the no circadian rhythm in the normal mouse embryonic stem cells, like here, like this. Okay. So in this case, not only the total system of the mouse embryonic stem cells. So mouse embryonic stem cells are derived from the inner cell mass of a blastocyst. So blastocyst is an early embryo, and then so of course early embryo early embryo does not care of the circadian rhythm. Just depend on the mother. Depends on the mother. Then so mouse embryonic stem cells has no circadian rhythm. Okay, like this. So even by the addition of sealed one, so no circadian rhythm here. But in this case, we differentiated mouse embryonic stem cell to the hepatic tissue, and then you can see the circadian rhythm of the albumin expression, like this. After the addition of a sealed one, like this, okay? And then also when he checked the other gene expression, I cannot read. So HNF4 alpha is a hepatocyte nuclear factor, and SREBP1 is I forgot. SRE binding protein 1. And ABCC2 is one of the representative transporter. So cyclic AMP dependent transporter. Okay? And so SHIP2 alpha 4 is also a metabolic enzyme. SHIP2E1 I have already explained, and SHIP2B10 also one of the metabolic enzymes. So this also so he checked many circadian rhythm related gene, and then he succeeded to make the circadian rhythm in the mouse embryonic stem cell derived hepatic tissue. Okay, and also check the circadian rhythm like this. So it's so very important to think about alternative methods of animal testing, because anyhow, so animal has a circadian rhythm, but in the general cell culture system has no circadian rhythm. But we introduced the artificial transcription factor gene into the cells, especially into the embryonic stem cells. And then so embryonic stem cells were differentiated to the hepatic tissue. So at that time, so I didn't explain. So here, this is the hepatic tissue. So red ones, hepatocytes, because producing albumin protein. And green ones are I forgot. CD31 positive cells. It means endothelial cells. So this tissue consisting of hepatocyte and endothelial cells at least. Then so we call the hepatic tissue. Yeah, so mouse embryonic stem cell derived hepatic tissue. Then so it's a cultured system, and then so we succeeded to make the circadian rhythm in the embryonic stem cell derived hepatic tissue. Then so it's one example. It's one example of the alternative method of animal testing. because this system has a circadian rhythm, and also it's a very functional. So this hepatic tissue has many hepatic functions like this. Producing albumin and also much and also he succeeded to indicate metabolic enzymes such as the CYP2A4 and the CYP2E1 and the CYP2B10. So if you use the primary cells and also if you use the cell line, hepatic cell line, such as the HepG2 and so on, and then maybe you can't observe these expression of a metabolic enzyme like this, and then so it's a very special. And then so, but so it's one example, but we added the sealed one periodically. Just for exposure time is for 30 minutes or 1 hour. We can control the periodical field. Now, so we succeeded. So I showed just a succession of circadian rhythm. It means a 24-hour periodical expression. But by changing the exposure time, we can adjust we can change the circadian rhythm period. So now 24 hours, but we can change to the 12 hours periodical expressions. And then so if you how can I say, so in order to check the circadian rhythm using this system, so you have to stay in the lab 24 hours because you have to take the you have to take the sample every 3 or 4 hours in a day, and so it's very hard work. But if you can change to the short period, so 12 hours, and then you can stay in the lab for 12 hours. Then so sampling is also maybe 2 hours, so every 2 hours. That's it. It's very convenient. And that's a very big advantage of the alternative method of animal testing. So in the animal testing also, you have to stay in the animal facility. And so 24 hours to take the blood at every 4 hours. And also we can't control the time of the period of time. So time of the periodical so circadian rhythm. And so just so you have to stay in the animal facility 24 hours. But in this using this system, so you have to shorten. We have to show we can shorten the circadian rhythm from 24 to 12 hours or 8 hours also. But now so it's quite difficult. Just maybe we succeeded to shorten the 12 hours. Just it's very convenient. And also using the microfluidic device. So we can add we can control the exposure time also. Just we have to set the program of the administration of a CO1 and also to take the samples also, and it's very convenient. Anyhow, so circadian rhythm is very important. So I hope you can understand circadian rhythm is very important to the for the diagnosis of the drug metabolism and pharmacokinetics. Okay. So maybe so it's almost finished, and yeah. So in the afternoon, I will explain another point. So it's difficult to culture cells as a normal cell origin and so on. Maybe I forgot. Maybe yes. Yeah. Yeah. So in the afternoon, I will start, and so in the morning session, I will so stop now, and then so shall I start at 1 p.m.? It's okay, 1 p.m. Okay. See you at 1 p.m.
It's done. It's done.
Okay.
Yeah.
Yeah.
We can start.
Okay, so let's restart. And in the afternoon, so I'll talk about difficulty to culture general, so culture cells as normal cells of organ origin. And so first, anyhow, so it's very difficult to culture normal cells from organ. Most commonly cultured cells are cancer cells. So, for example, in the liver, we have a variety of cell lines. For example, HepD2. HepD2 is also derived from the liver cancer. Cellular carcinoma, hepatoma. And then so HepD2 lost several hepatic functions. So liver cells are very special cells. General other cells in the organ, maybe one sort of cells has one function. But hepatocyte is very special. Hepatocyte has a lot of functions, specific hepatic functions. So hepatocyte have a variety of metabolic enzymes, and also producing the albumin and other proteins also. And also hepatocyte have a function to detoxify the toxin, and many functions. And so, but HepD2 has been established from the hepatoma. It's a liver cancer. And then so generally cancer cells back go cancer cells go back to the immature stage, and then so hepatoma cells also have so lost many hepatic functions. Then so hepatid2 lost metabolic enzyme function and other functions also. And also other cell lines, so Hu7 also are very convenient cells because it's susceptible for HCV infection and so on. But unfortunately, Hu7 has also lost several hepatic functions also. So, for example, so in order to make the artificial liver system, of course, it's a replacement for real liver, and then so artificial liver system needs all of hepatic functions, and then so engineer they developed many types of reactor to culture hepatocytes, but unfortunately they didn't have a good hepatocyte cell lines. Then so it's quite difficult, and also so of course they could prepare the primary cultured cells, so hepatocytes, from the not only from the human, from the porcine and porcine is a pig liver, and bovine cow liver and so on. But unfortunately, primary hepatocytes has also lost many hepatic functions, and also primary hepatocyte cannot proliferate anymore. Then so it's very difficult. Anyhow, so in general, most primary cells cannot survive in culture. And most primary cells from organs do not proliferate. Now, most primary cells from organs lose their original physiological functions. And then so, first problem, for the first problem, most primary cells cannot survive in culture, but now we developed several media with compositional or other so adequate extracellular matrices. It's a scaffold that allows cells to live. So, but not enough. Then so we are now also continue to develop the medium and adequate ECMs. So ECM is I have already explained in the morning. So extracellular matrices. It's a scaffold. So general cells have to adhere to something. So something we call the scaffold. Okay, scaffold. Always in cells have to adhere to something. So it's a scaffold. Then so such as collagen or fibronectin, and laminin, and so on. It's very important. Then so for example for hepatocytes, we used Williams E medium and also we use collagen as a scaffold. But not enough. But anyhow, so now we can culture the primary hepatocytes a little bit for a long time, and for a long period. And then second, most primary cells from organs do not proliferate. So it's a very big problem. So the primary hepatocytes and also other cells, so in organ. So these cells have been already matured. So matured cell cannot proliferate. Immature cell can proliferate. So cancer cell, it's like an immature cell. And then, so that's why cancer cell can proliferate. But the mature cell, for example, so skin epithelial cell, already mature. So this cannot proliferate anymore at the moment. But now we can, because we have already developed a good way to proliferate the skin epithelial culture. But anyhow, so but other cells, for example, so cardiocyte is heart cells cannot proliferate anymore. So and the liver cells and also pancreatic cell, pancreatic beta cells also cannot proliferate anymore, and other cells also. And so then, but now we can prepare the stem cell from each organs sometimes, but and also we have embryonic stem cells or iPS cells, and also other anyhow, so mesenchymal stem cells. So we can use the stem cells to differentiate into the matured cells. So now we have established. Then, so 10 years, around 15 or 20 years ago, we used animal cells. But now we have human ES or human iPS cells, and also human mesenchymal stem cells. We can use. We can get these stem cells, human stem cells. And then, so of course the final purpose to be applied for the clinical in human. And then, so now we can use the human stem cells to differentiate into the human mature cells. Okay, and third one, most primary cells from organs lose their original physical function. It's also a very big problem. So now, just I can suggest to consider its microenvironment in the organ or tissue. So I have already explained liver microenvironment in the morning, and then so now we are mimicking the liver microenvironment for the establishment of hepatic tissue culture model. And then so other organ or tissues also, maybe so you can consider their microenvironments in their organs or tissues. That's my suggestion. Okay. And then so total is now so we have several good adequate media and adequate ECM for the cells, primary cells or differentiated cells also, and also we can use stem cells, for example, such as ES or iPS cells, especially especially human iPS cell or human mesenchymal stem cells and so on. To differentiate into the purpose cells, and also we are now mostly we know its microenvironment in the organ tissue, because we have done many animal testing and also clinical from the clinical information. And anyhow, so let's move back to the liver. So this is these are primary cultured hepatocytes from the DS-red fluorescent gene expressing transgenic mouse liver. And that's why these cells have a red fluorescence. Okay, and so you see it's quite difficult, but you can see. So this is a single hepatocyte. You can see the two nuclei. Okay. And so generally, matured hepatocytes, most of the matured hepatocytes have a binucleus like this. So they are also same. Okay, and but unfortunately, so these primary hepatocytes cannot survive for a long period, and cannot proliferate anymore, because it has been already matured. And of course, there are many trials to add some growth factors, such as EGF and FGF and so on, but no function to proliferate anymore. So and also it's very difficult to maintain the hepatic functions also. It's quite difficult. I can give yes. So maybe tomorrow I'm going to talk about potential, so differentiation potential over in the ES or IPS cells. Then so I will talk about the differentiation over IPS or ES cells to the liver tissue. And then so I will talk about more detail. And anyhow, so that's today that's an introduction of the limitation of the cell culture system. And also, mammalian cell has and then so now a little bit move on molecular biology. So there are so many differences between mammalian cells and the bacteria. And so now we're looking at genomic DNA. So mammalian cell has a linear chromosomal DNA. But on the other hand, bacterium has cross-cycler chromosomal DNA. So like this. So bacterial genomic DNA normally also circular form. And the mammalian genomic DNA is linear. It's completely different. And normal, and also normally, so normally, replication site, so origin of the replication is a single in the bacterium, but sometimes now also there are several origin sites in also bacterium genomic DNA, but normally also single site for that. And then so replicate starting here, and then synthesized and from the 5' to 3', and then so reach to the start point, and then so no and also normally so DNA polymerase require primer, you know, and then so normally RNA because discrimination of genomic DNA or primer is very important. Then so primer, and so reach to the start point, and then the RNA primer should be degraded, and then connect to the start of the DNA synthesized points, and then so it's okay. The mammalian, in the mammalian genomic DNA, so there are many replication sites, but especially, so let's look at the terminal here, okay? So length remains, so length of the genomic DNA, bacterial genomic DNA, it's a circle, and then so length remains, so length of the genomic DNA remains the same after replication, of course. On the other hand, in the mammalian genomic DNA replication, the length is reduced after replication. Okay, it's a very big difference. And then, so this is a chromosome, human chromosomes like this. So here, it's a telomere. It's a terminal region of a chromosome here. Okay? And so anyhow, so there are many sites of the replication, so origin of the replication in the genomic of in the mammalian genomic DNA. But now we'll just look at here, so terminal, okay? So anyhow, so DNA polymerase requires primer, usually RNA, and then so because the initial double-stranded Watson-Crick base pairing prone to error. Then so they are corrected exactly as the DNA once synthesized has proceeded, and except for the terminal. So here is they can be corrected, but except for terminal. So terminal is it's impossible to correct anymore. And then so but RNA primer should be used here also. And then so and then so DNA replication is finished, and then so maybe RNase H activity enzyme digest these RNA primer here. And then little bit shortened. Okay, and then so it's shortened, and then replication again, and then shortened next here. And then finally, so chromosomal DNA becomes shorter and shorter. It's a very sad system in our mammalian genomic DNA replication. So gradually shorten. So it's senescence. So senescence, do you know senescence? It's aging. So that's why so primary cells, so matured cells cannot proliferate anymore on the culture system because already so aged cells, matured means aged cells, and then so primary cells have already shortened chromosomal DNA. Okay, that's the main reason. And but you know, so cancer cell can proliferate, and then so of course we have a special system to add to stop the shorten the chromosomal DNA. So and then so here telomere is a terminal structure of a eukaryotic chromosome with the animal species specific repeat sequences. Okay, it's so it means no meaning, so non-coded region for the protein. Okay, and then we have a telomerase. It's an enzyme. This enzyme so is so reverse transcriptase and RNA strand complementary to telomeric repeat sequence required for maintenance of telomeric structure. So anyhow, so we have telomerase. So telomerase add some sequence to at the end of the chromosomal DNA. So to stop the shorten. Okay. So cell divided shortening of telomere chromosome DNA leads to aging. So we call cell senescence. On the other hand, cells divide and then no longer shorten of telomere of a chromosomal DNA leads to stop dying. So it's cell immortalization. So does not die. Okay, immortalization. And then immortalization. So telomeres do not shorten. So because the telomerase is active, and telomerase shorten the telomere lengthen again. In normal matured cells, telomerase does not work. But in germ cells or embryonic stem cells, I'm going to explain what the embryonic stem cells are in tomorrow, and also iPS cell. Also, I will explain tomorrow. Anyhow, so in germ cells or ES cells and iPS cells, the telomerase is active. And for example, it's a little maybe tomorrow I will explain again. So after the by transplantation of embryonic stem cells into the body, and then so benign tumors forming, we call teratoma, the benign tumor. But anyhow, because embryonic stem cells have a very high active telomeres. And in cancer cells, of course, the telomeres is very active. Then, so that's why cancer cells can proliferate rapidly. Okay. And I will. So I'm going to explain telomerase reaction a little bit. Maybe you have already learned these events, right? I think. And then so, if you are not so interested in this system anymore, and then maybe you will forget this. So that's a little bit. I will explain shortly, briefly. Here, maybe here, just here. So this is the terminal of the chromosomal DNA, like this. Then, so since it's there, so here it's the template of the DNA. Then, so start replication is start here normally, but anyhow, so it's shortened. And then, so telomeres are already, so telomeres is just a complex with protein and also RNA. So the telomerase RNA here, then telomerase RNA recognize the terminal of chromosome TTG here like this. And then so since it's, and then so along this template, and then so this terminal is prolonged. So synthesized again along this template here like this. Then so TTG add the GGG. So it's a very special sequence, and then so TTG. Then move to the again. So this sequence also includes the TTG again, and then so this sequence is skipped to this, and then synthesized again, and then so this chromosomal DNA is lengthened like this. So template DNA, so telomere DNA contains the guanine-rich sequence. So normally 6 to 8 base pair long repeats of sequence. Maybe so this telomere sequence is very long, and so kilobase pair order in human, and in a mouse more longer. And then so this is a 3' overhang region. It's cap, and then so lengthen 3' like this. And then so based on this information, then so we invented special technology to culture the normal cells eternally. Then so first one is a genetic engineering method, and another one is cellular techniques in culture. And then so first I will explain so genetic engineering method. First one is so based on this previous explanation. Then so overexpression of telomeres. And then so first telomerase expression vector should be constructed. And then this telomerase expression vector should be introduced into the normal cells. Then, so these expression vector would be integrated into the chromosomal DNA, and then so we can get the cell lines. So by expression of telomerase, so we call H tart. H tart is H is a human, and the tart is a gene name of gene of telomerase. And also another one. So overexpression of oncovirus genes. For example, SV40 large T antigen, HPV, E6, E7, and EBV. And so oncovirus is oncovirus means so after the infection of this virus, then so a tumor appear in the body. So SV40 is so simian virus 40. It's so monkey virus, and then but large T antigen is very special. I will explain later. And also cell cycle is regulated by the RB or P53, and then so these we call the cancer suppressor gene, and then so just by making the knockout of these genes, then so and then to be immortalized. Okay. Then first, just a moment. Telomerase is just very strange. And so second one is cell fusion with tumor cells. I will explain later. First, I'm going to explain the SV40 large T antigen introducing. So introducing and then so induce the immortalizing. Okay, and then so first, so this one is a primary cells. Primary cells prepared from the normal tissue, normal organs. Then so I have already explained many times, and so primary cells are very low proliferative. So primary cells have low proliferative potential, but now we want to culture, expand these primary cells, And so first, SV40 large T antigen expression vector was constructed, and then so this expression vector would be introduced into the primary cells here, like this, by, for example, electroporation or something. Then, so SV40 large T antigen replace the P53 or RB. So these are so cancer repressor gene. So, but these molecules regulate the cell cycle speed. Okay. Then so, but SV40 large T antigen suppress the function of RB and P53. And then so this cell lost the brake for the cell cycle speed. Then so to enhance the proliferate Okay, and then so anyhow, so after the introduction of the SV40 large T antigen expression vector, and then so they got high proliferative potential like this. It's a very smart idea, and then so there are many cell lines. For example, COS7 COS7 cell line. It's a very convenient cell, mammalian cells. It's a monkey cells. Maybe rhesus monkey derived cells, maybe. But anyhow, so COS7 is very famous, and maybe most of researchers, they are using the COS7. I also use COS7, and then so COS7 has also SV40 large T antigen. And so it's very useful. And the other cell lines, I forgot the name. But anyhow, so there are many cell lines carrying the SV40 large T antigen. And very convenient. But these cells having the SV40 large T antigen just for the culture, or just for the production of some proteins, and also for the virus infection model. Then, but so it's quite difficult to use these cell lines having the SV40 large T antigen for the alternative system of the animal testing, because completely different from the normal cells. Of course, so they have the SV40 large T antigen, and then so proliferation, so cell cycle system also very abnormal, like tumor. Then so these cell lines cannot become the alternative models of the animal testing or human testing. And the next, cell fusion system. I forgot. Yeah. So cell fusion method is normally by these three methods. So first, so maybe third one is not so popular. So second one is the most popular, polyethylene glycol, using a polyethylene glycol. But the first three are Sendai virus infection. So Sendai virus, formal name is hemagglutinating virus of Japan, HVJ. So because Japanese scientists discovered this virus, and then so Professor Dr. Ishida discovered discovered HVJ, and then so he worked at Tohoku University Sendai, and then so that's why Sendai virus called the Sendai virus, and then so Dr. Okada discovered the cell fusion action by the infection of HVJ, HVJ. And then so mechanism are like this. So here cell A and cell B, and then HVJ infected here. Then so HVJ bound to cyanic acid residues in all residues in the cell membrane. Then so fused. It's HVJ is a mediator to fuse the cell membrane like this. We call this situation heterokaryon, and then finally nuclei also fuse each other, and then to become the single cells like this. Then synkaryon, we call synkaryon. Heterokaryon, so synkaryon become the synkaryon through the heterokaryon like this. Of course, you can understand. And then synkaryon cells, so fused cells are divided like this, and then so we can get cell proliferation like this. And the polyethylene glycol also same role, and so polyethylene glycol its structure is like this. It's a very simple structure, and then so polyethylene glycol has a very big advantage. It's a non-toxic, so bound to hydrophobic molecules, and so non-hydroionic surfactants and emulsifier in the cosmetics, and bound to a protein drug, inhibition of proteolysis, and also to make the bigger size of the molecule. For example, a very famous one is so PEG interferon. Interferon is a cytokine, one of the cytokine, and so interferon has plays an antiviral effect. Then so now maybe they don't use the interferon, but maybe 10 years ago, no, 5 years ago, so HBV, so B-type hepatitis virus infected patient, or C-type hepatitis virus infected patients. So they had received interferon treatment, because interferon plays a crucial role to be an antiviral effect. But just the injection of the interferon. But the patients had to go to the hospital daily to receive the interferon infection. Then it's a very bad, low QOL, so quality of life. It's dramatic, so very, very bad. Then, but some pharmaceutical companies developed the PEG interferon. So PEG bound to the interferon, so this one. Then so interferon is not a protein, so it's a peptide, very small molecule. And then so this small molecule is immediately excreted at the kidney because it's very small. But pegylated interferon become the large molecule. And then so large molecule you cannot be excreted at the kidney naturally. Then so it's very convenient, and also PEG is non-toxic, and then so it's very convenient. And then yeah, drug at large. Yeah, it's so inhibition of discharge into the urine. Okay. And then so PEG also same role as HVJ. And then so PEG bound to the cell membrane molecule like this. That it's a mediator to the cell fusion, for the cell fusion like this. Yeah, so like this. Okay. Yeah, heterokaryote and nucleus fusion and missing carrying. Okay, and the cell fusion. So it's also a very smart manner, but this manner also are not so good for the normal cells. Just so, for example, so this one is a normal cell, but normal cell each other fused, or it's just a normal cell, so cannot proliferate anymore. Then so counterparts should be tumor cells. Then so tumor cells proliferate and can proliferate, and the fused protein also can proliferate. And also some so can maintain the specific function also. But anyhow, it's like a cancer cell, so not normal cell. And so cannot become the alternative method of animal testing or human testing. But it's very useful for the antibody producing cells. So plasma, so fused plasma cells producing the monoclonal antibody, and with other tumor cells. It's very convenient. Anyhow, it's just a moment. Why this here? Just a moment. Just a moment. Okay. Yeah, and then so anyhow, it's quite difficult to maintain the hepatic function in the primary hepatocytes like this. So hepatic function is dramatically decreasing day by day. You can see like this. So it's quite difficult. Then so how to do it? So how to proliferate? So we have. So I introduced several ways. First, we introduced the telomerase expression vector into this primary hepatocytes. And then so we maybe primary hepatocytes can proliferate eternally. And the second one, of course, we can introduce the SV40 RGT antigen into these primary cells. Also, hepatocyte, the primary hepatocyte, of course, changed, but anyhow, so proliferate, can proliferate. And the next one, third one is by cell fusion. I mean, primary hepatocyte fused with hepatoma cell line, for example, HepD2. It's also possible. So which one is the best? Or of course you can find another way. But if you want to expand my primary hepatocyte, how to do it? So maybe which is the best? Which is the best? It's a very difficult. So,
Can you repeat the question again? Because I didn't get that. Teacher, can you repeat the question again?
Okay.
I didn't get it.
So, primary hepatocyte prepared from the liver. In this case, so mouse liver, but of course we can get human primary hepatocytes from the human being, so human liver. So at this time, so unfortunately, this donor, this donor already was killed or died. Okay, and then, but we can't expand. Expand is so means expand primary human hepatocyte. Expand. So it means primary hepatocyte cannot proliferate. Okay. And then also lose the hepatic function also after the culture. But anyhow, so we want to expand and we want to maintain the hepatic function in the culture, primary hepatocyte from the prepared from the human liver. So, do you have some idea?
Maybe keep the telomerase in the cells work,
Mm-hmm.
and you can introduce SV protein.
Mm-hmm.
Yeah, I think to prevent these from die. Yeah, and also to give us the microenvironments like the normal cells in vivo.
Okay, so I agree with the last one. Okay, but so I explained to introduce the telomerase expression vector or SV40 large T antigen expression vector to proliferate for the proliferation of the primary cells. But I told you so these are not so good because SV40 large T antigen is a very abnormal gene because comes from the SV40. It's a virus. Then so we don't know. So we know just large T antigen suppresses the RB or P53 function only, but we don't know the other function, large T antigen, and then that's a very big problem for applying the clinical treatment. It's very dangerous. Also, SV40 is not a human virus, so monkey virus. And so we don't know what function it has. Okay, so SV40 cannot infect human being, so just so infect monkey. And then so I don't know the disease in the monkey, but anyhow, so it's maybe after the infection, a monkey maybe have some tumors or something. But anyhow, so and then so maybe so we can now we understand maybe the mechanism of the tumor formation in the monkey after the infection of SV40. But I don't know the other mechanism. So large T antigen can bind a very important gene in the human, so important protein in the human or not. So it's very dangerous. And and also affect some physiological functions also. And then so it's not so normal. And also by introducing the telomerase gene, so expression vector, also maybe telomerase, of course, plays a lengthen the genomic terminal of genomic DNA. That's I think it's one function, only one. So one of the function maybe. So telomerase, of course, we know just only the maybe the just lengthen the genomic DNA. It's okay, but we don't know other function. And maybe affect some effects also. I don't know, but and then of course maybe the by cell fusion is a very bad manner to make the to reach our purposes. And then so it's not so good. And then so now then so today so almost finished because maybe I have to explain detail more, but so almost finished maybe. And then so back to this slide, and so we have for example, just a moment. Yeah. Anyway, so it's a very difficult. And then so now we can use human ES cell or iPS cells or mesenchymal stem cells and so on, because these are normal cells. And also we have already some data after the implantation of the human ES or human iPS cell derived tissue in the patient, into the patient already. And then so we know these cells have so we know the fate of these differentiated cells from the human iPS cell or human human ES cells. Then so tomorrow, mainly I will explain the human ES first, and then so next human iPS cells, and so on. Then, but these are also just most of them so just so differentiated to the one sort of the cells. So, for example, they have already established the differentiation system into the hepatocyte from the human iPS cell or ES cells. That's it. And then so just so after the its implantation is okay. So after the implantation, these cells can survive in the human body. But the purpose of this lecture, we are now considering the alternative method of the animal testing or human testing. And then so it's still quite difficult to make the culture model using the regenerative medical technology. So we can differentiate human iPS cell into the each cell types of the organs, but cannot keep the function on the culture. So these differentiated cells should be immediately transplanted into the patient. And then blue one. So I will move on the blue one. So blood circulation cannot be reproduced in the culture system. So blood circulation means I have already explained in the morning. So erythrocytes, of course, erythrocytes exist in the blood circulation system, and erythrocyte bring the oxygen and supply to the peripheral tissues. So it's very important role, and also other cells of course lymphocytes, so such as T and B and the macrophage and so on. These cells also exist in the blood circulation, and also some signaling molecules also contained in the blood. And then so, for example, distance between the intestine and the brain, very, very far, you know, brain here, intestine here. But microbiota in the intestine affect the brain disease. So already, so there are several papers. Then so maybe using the animal, maybe so we can show the mechanism of these, so these cells. So, event, maybe. But now we have to consider alternative from the animal model. And then so we don't use the animal model. Then so just so culture the bacterium, it's okay, and just culture the neurons, it's okay. But it's a very difficult to reproduce or reconstruct the total system. How to connect to each other, and it's very difficult. And also, so I have already explained now microbiota, and anyhow, so consistency of the indigenous bacteria cannot be achieved in the culture system also. And then, so last day, so Wednesday, I will talk about one solution against these problems using the microfluidic device. Okay, then so now general culture cells, there are many problems, but maybe we can overcome these most of these problems by using the microfluidic device system. And also not only the using the microfluidic device, and also using the regenerative medical technology by using the human iPS cell and sometimes the human ES cell also, or mesenchymal stem cells. So it's a very important. Anyway, so today I have around 40 minutes, but I prepare all of them slide, and so maybe I'm going to receive some questions. Okay. If you have a question, so please raise your hand.
I have a question about cell fusion. I have a question about cell fusion. Sorry. Just we know that cell fusion is normal cell fusion with the tumor cell, but I think for the future maybe they have double materials in cells, like double DNA. The number, like the number is much more than the normal cells. So I wonder why it can work, like the fusion cells can work.
Okay, so first we are so fused cells, of course, at the syncarion state. So chromosomal double, number of chromosomes double, but divided. And sometimes maintain the double number, but sometimes crossed the chromosome. For example, I forgot to tell you. So this cell fusion system, just same animal, so deriving from the same animals, and then so we succeed to keep the chromosomal number of chromosomes. Then, so for example, to produce the mouse antibody. Then, so mouse. So we prepare the mouse plasma cell, and also the tumor cells, also so mouse cells, then fused after the fusion. Then so it's okay, and we can keep the number of chromosomes. Maybe sometimes double or half the same, because most of chromosome sequences are almost same, and they can recombinate. They can recombinate each other, and it's okay. But for example, so also we can make the fusion cells between the human and other animals. Then so human chromosomes lost, and then only one chromosome remain after the fusion. And it's also a very useful for the location or location of the specific gene, and because we can recognize which chromosome number and, from the shape of the chromosome, and it's also safe. And so it's useful. But anyhow, so it's quite difficult to keep the chromosome, so native chromosome. It's quite difficult. But cell fusion from the same animal, it's possible. Then so. And then so sometimes double or sometimes maybe in the, back to the single sometimes maybe, but anyhow, so I don't know exactly, but just a moment. And also there are several fused cells in our body also. For example, muscle cells. These are so fused cells. And then so of course muscle cells can survive. Yeah. And but muscle cells maybe so cytoplasmic area fused, but they have many nuclei. I think. Yeah. I have maybe I have better confirm this, but anyhow. And also many, and also I forgot. Yeah, I have a specific slide for this question, but today I don't have. So maybe tomorrow I'm looking for that slide, and so I will show you tomorrow. Okay? Yeah. Okay, good question. And other? Okay. So please find so other student.
I have a question about if today we are using a normal cell fused with a cancer cell, this should be a hybridoma. But this hybridoma, I always have a question about when we are fused, the cancer cell that we are derived from the benign tumor cell or is a malignant tumor cell? Yeah, and another question is, in our conventional wisdom, we all thinking about the cancer. It's kind of a disorder of the gene. So my question is the hybridoma, this precision, this progression is by the genetic disorder, or it is because I have some implication that maybe it is caused by the metabolites. It's kind of a metabolite disorder because in previous I have saw one paper that it just put the normal cell nuclear put into the cancer cell, and it was to become the cancer cell. The gene inside the nucleus has changed to be more like cancer. But if we put the cancer nucleus into normal cell, that it will not to become the cancer cell because it will to repair the some of the disorder of the gene. So I was always wondering about the hybridoma. This is very special because we cannot to identify whether it was a genetic disorder or something, a metabolite disorder during this progress.
Yeah, it's possible. So I don't know exactly because hybridoma. Yeah, just we can observe the phenomena just on the culture. Then so no, if there are transplant hybridoma cells into the animals,
Because we are always using to prepare the vaccine, and we just to put some of the plasma cell into the mouse and become the hybridoma. But always the background of some of the knowledge that we always confuse because we don't know about why they become the hybridoma.
Yeah, it's a very interesting point. Yeah. You can do it. Maybe greater. Okay.
I think so. I don't know.
Yeah, it's a very interesting point. Yeah. Okay. Other student? Okay.
And here you talk about the microenvironment, and I wonder why, how to define the molecules in the microenvironment.
Very severe question because it's quite difficult to mimic the whole microenvironment as organs one. Maybe very difficult. Now, so just we are trying to reconstruct the structure or histologically mimicking the, for example, liver tissue structure. And so we are now focusing on the cellular polarities, and so to make the apical, basal, and lateral base. But of course, it's quite difficult to make the real, so really same structure. It's very difficult. And then I think depends on the purpose. Depends on the your purpose. Yeah, so it's okay or not. Yeah, but anyhow, so major difference between the culture and the body. Anyhow, so our cells in the body prefer the aerobic condition. Always we need oxygen, always, because erythrocyte can supply, always supply oxygen to the peripheral tissues. It's so aerobic condition, except for the inside of the intestine. It's already outside of our body. Yeah. So in the bowel, so it's completely anaerobic, and then prefer anaerobic bacteria prefer this condition. But of course, the epithelial cells of the bowel, of course, need oxygen, and so blood vessels lead to the epithelium of the bowel, so large intestine. Then it's okay. But anyhow, so we need oxygen. So we prefer aerobic condition. But unfortunately, in the culture condition, so it's quite difficult to make the aerobic condition. Always anaerobic, normally. And then, so maybe last day I will introduce some technology to supply the oxygen to the in the culture condition. So, for example, they use the PDMS, is polydimethyl siloxane. So it's a silicone. It's like a silicone, one of the silicone. And then so it's gas permeable, and then so oxygen can pass through the this device, and then so to supply the oxygen reach to the cell culture medium. But also it's just oxygen. Maybe also. Oxygen carried by the erythrocyte. Maybe so chemical form is also a little bit different. Maybe a little bit ionized and something, and so it's also quite difficult. Yeah, and then so I think yeah, it's very severe. So. And so, of course, I have many presentation lecture about our device, and then I'm afraid of receiving your question. Is it really mimicking the microenvironment? Yes, some part of them, but not all of them. It's quite difficult. Yeah.
And I think like the medium to culture the cells to mimic the environments will be difficult.
Yeah.
But
Yeah. Yes. But now, so we have known the composition of the serum, and serum means so blood, composition of the blood, and then so we can prepare or we can get similar composition liquid as blood, and so it's almost achieved, maybe, but. And also now some of my friends, they are developing the artificial erythrocyte also. And then so maybe, but efficiency to supply the oxygen is still very low. Then so it's quite difficult and also very expensive. And so I can't use these artificial erythrocytes. But anyhow, so I'm waiting for prices decrease. I'm waiting, but anyhow, so yeah, very difficult. Okay, other questions, comments? It's okay. You have no? Okay. And other? Okay.
Yeah, I wonder, do you use the cell which can proliferate in your microfluidic cell?
Sorry,
did you use the cell which can proliferate in your microfluid system, or you use primary cell?
Yes. So depends on the situation, or depends on the purpose. And so Wednesday, I will introduce several types. One is one cells derived from the mouse embryonic stem cells, and another one is from the human iPS cell, and also sometimes we are using the cell lines also. Of course, honestly speaking, so cell line is very useful and very convenient and very less stress. Yeah, and very economical is all economical. But using the human iPS cell, it's much stress because medium, very expensive, and we have to wait 1 month or 2 months to get destinated cell. We have to wait. So not only for me, so graduate student also. So they have tried to differentiate human iPS cell into the, for example, brain tissue cells, and then so they have to wait for 2 months, for example. Then 2 months, but so 1 month it's okay. Well, so how do your cells? Oh, it's okay. Now so I'm waiting for one month more. Then tomorrow, for example, I met him. Then his face is very bad because contaminated. So 1 month. So he spent maybe, for example, so maybe million Japanese yen already, and then contaminated. So very much stress. Yeah. Sometimes happen. So I don't care. But yeah, it's very much stress. Yeah. Sometimes. And then so now my opinion. So just we are going to buy these cells derived from the human iPS cells. Very expensive, but I think if you try to differentiate human iPS cell into these cells, maybe you need 5 million Japanese yen or something. But just maybe I have to pay the 1 million Japanese yen. and also it's more stress, and then it's so yeah. Firstly, so looking at the price, the list, price list, very expensive. 1 million, 1 million, yeah, for one vial. Not one vial, so 10 vials. Very expensive. But so calculated, so yeah, PhD student, so calculated, and then more. But I can differentiate the human iPS cell to the brain tissue cells. So maybe I need just 0.1 million. It's enough. But so he spent 2 months, and before 2 months, he contaminated, and then he tried again. So he spent 0.1 million yen again, and then he failed, and then so try again, and then so finally he succeeded, but he spent 1 year. Yeah, I think. But of course, so PhD students, they receive the lecture, and then so after they feel this field very interesting, and then that's why they wanted to join in my laboratory. And then, of course, they want to try to differentiate the human iPS cell into the these cell lines, these cells. Yes, but now so it's not a study. It's so it's possible to to buy them. But PhD students, so they want to try anyhow. But my opinion is, don't do it. Just buy it. So I can pay. It's a little bit gap between my opinion and the students.
Because as I know that traditional ways to make the iPS cell to be a different kind of cells that this procedure is very hard and very unstable. So it is very likely that you get the failure when you are trying to make the iPS cell to be like brain cell or hepatocyte cell. Yeah. So if me, I would try just to buy the cells that I want to use. I will not try not to use the iPS cell to differentiate.
Yes. Yeah. You're good part. But
it is an experience.
Yeah. But so But I can imagine. So yeah, why they think about it. Yeah. Because when I was a PhD student, so I produced interferon gamma knockout mice. At that time, so before starting this project, American group succeeded to get to produce the interferon gamma knockout mice. And then my teacher, so my boss, he told me to stop this project.
Just going to buy it.
Yeah. But I wanted to produce it by myself. And then always discussing, and then afterwards, my boss permitted to produce interferon gamma knockout mice. But the construction is a little bit different. So my interferon gamma knockout mice, not only the knockout, and also the beta-galactosidase expression by a replacement of interferon gamma. And then it's a special. And then succeed. Then Also, I checked by myself everything, and I made histological examination also, and many things. And, so it's a very good. So it's a very good. And but finally, and then so my interferon gamma nucleus now completely occupied in the world. So because we have a very big group in Japan, because the interferon researchers, many very big population in Japan, and then so they used my interferon gamma, and also I have a European and American friend also, and they used my interferon gamma. Then also we have a very good breeding system, and then so it's okay. But anyhow, so this project, I mean, so to differentiate the human iPS cell to the nerves tissue and or intestine. So now so we can buy. It's enough, I think. We want to look at more.
More further.
Yeah, well, yeah,
mechanism.
further. Yeah, further.
It's not
Yeah,
just always focusing on the cell differentiation.
Yeah, but anyhow, so they have heard my lecture, and then so always so firstly so I introduced these basic points, and then so of course it's very easy to understand the introduction, and then so it's very interesting. It's a very simple. Yeah. Okay. And other questions? Okay. Almost. Yeah. Almost time. Yeah. Okay. Tomorrow.
Teacher, some student asked online.
Sorry. Please read it because too
Okay.
small. Too small.
She say she wondering if it is possible to activate the polymerase to maintain the function of primary cell.
She wondered how did the telomerase help to maintain the function of primary cell?
Yes. So I'm sure, but anyhow, so primary cells, maybe early period of the primary cell, of course, have a function, but it needs some period, long day to get clone after the introduction of telomerase expression vector. So maybe during this screening period, these functions unfortunately lost, I think. So maybe you can imagine. First, of course, firstly, so we have a telomerase expression vector, right? And then and also we got primary cells, for example, primary hepatocytes. Then, for example, 10 million cells, and then this expression vector should be introduced into the primary cells by electroporation. And then, so efficiency of the introduction, so introduction efficiency is not so high, maybe 10% or something. And then, so we have to culture these cells anyhow. So maybe for 7 days or 10 days. So we have to wait, proliferate again. Then, so if you are lucky, maybe you can get one clone to have a good function, but another one, of course, no function already, because so during the culturing, so we spent already 7 or 10 days, and then so function dramatically decreased, and it's quite difficult to get this functionable clone. It's quite difficult, I think. Then so sometimes, for example, TWNT4. TWNT4 is a sterate cell line. It's a sterate cell is one of the member of hepatic tissue, sterate cell. So accumulation of vitamin A, maybe, I forgot. But anyhow, so function. And then, but it's quite difficult to culture the primary sterate cell on the dish. And then, so one researcher, he tried to introduce the telomerase expression vector into the sterate cell. So then, so finally he got sterate cell line. But this function, yeah, it's a functional. Yes, very convenient. He's very lucky. But the problem is very high proliferation rate. Also a very big problem. Because the sterate cells, it's normal, it's very slow proliferate, or stop the proliferate in the liver. But when the liver change to the fibrosis, and then sterate cells are activated and to proliferate, and maybe
it's all battery.
Maybe and so I forgot what I told. So when the liver change to the fibrosis, and then so sterate cell is activated and rapidly so proliferated. And so that's almost the same situation as this sterate cell line carrying the carrying the telomerase expression vector. Then so I think it's very difficult, just so difficult to keep the function and can be to be able to proliferate by using the introduction of a telomerase expression vector. there other question? No? Okay. So tomorrow I will explain back to the chapter back. So tomorrow I will introduce embryonic stem cells and iPS cells, and also finally I will introduce organogenesis or organoids, or so organogenesis and organoids. So organogenesis is development of organs embryologically. And organoid is artificial precursor or organ. It's now a very modern technology, organoids. And so also we are using the organoid cells for resource of our microfluidic device. Okay. Okay, tomorrow and then, so Wednesday I will introduce a microfluidic device, mainly our device. And also maybe I will show you video how to operate this microfluidic device. Okay. Okay, today that's all. Okay, thank you. Thank you for your attention. Oh yeah, and okay, just a moment. Yeah, maybe after back to my hotel, and then so I will prepare. Okay. Yeah. Maybe so. Maybe I prepare. Yeah, before. Sorry, before 10 p.m. Okay.
线上课，还可以线上参加。
对，
好爽哦，在家里就好了。好远。
声音他们会听不太清楚。
那就对了，是没错啦。
真的是嘉义大学